Hey everyone, welcome back to another tutorial video in which today we're going to be going through some of the example workbook questions that we're seeing in week 2 as we introduced amino acids. So if we have a look down here at our first question, it's saying draw the structure of glycine, aspartic acid, and lysine. Now these structures it's saying at a pH of 7, so neutral. So underneath that, we then need to write the overall charge. So this is a bit of a two-part question. So let's start with the first part in terms of just drawing the overall structure. So remembering that when we're looking at our amino acids, they all share the same backbone. So what I mean by that is that they will have the quaternary ammonium group, they will have the carboxylic acid group, and the hydrogen. So looking at our amino acids, again, they all share the exact same backbone. So all you need to properly remember is the R group, because that's the big thing that changes across all 20 of our amino acids. So let us start off then with glycine. So glycine is the smallest and arguably the most simplistic of the amino acids, in that its R group here, its side chain, is simply a hydrogen. Now, looking again at my backbone structure here for my amino acid, we need to have a look how it would be at a pH of 7. So at a pH of 7, what we would see is that our quaternary ammonium group, our carboxylic acid group, can change in terms of their ionization. So whether they have a hydrogen attached to them or not, and a subsequent charge. So looking at a pH of 7, if we are looking at our quaternary ammonium group, this is quite alkaline. So we are not going to see this become deprotonated. This is actually going to stay as NH3+. Now, looking on the other side, looking at our carboxylic acid over here, what we are going to see with this one is it's going to lose that hydrogen because this carboxylic acid, the hint's in the name, it is acidic. So at a pH of 7, this is going to be O-. Now, let us have a look at aspartic acid or aspartate. So again, we want to draw out our backbone. Once the backbone is all drawn out, we can then add the R group. So with aspartate or aspartic acid, what we are looking at here with that R group is that there is a methyl group and then a carboxylic acid. And our lucky last one is lysine. So again, I'm going to be starting this by drawing that amino acid backbone. Now for lysine, the R group here is going to be 4 methyl groups, followed by a ammonium group, like so. Now we need to then double check our R groups. Are they appropriately protonated or deprotonated at pH 7? So when we're looking at aspartic acid, again, it's a carboxylic acid here, so that would be deprotonated at pH 7. And with lysine, we have this quaternary ammonium group down the bottom here, and that would be protonated. Now, underneath the structure, we then need to write what the overall charge is. So for glycine, what we need to do is essentially, or for all of them really, we need to look at all these ionizable groups. So with our quaternary ammonium group on the left here, that is going to be +1 in charge, which we can again see over here. Our carboxylate, that is going to be a -1. And because this R group is just a hydrogen, it is non-ionizable. So that means that the overall charge for glycine is 0. It is neutral. Now, looking at aspartic acid, our quaternary ammonium group, that is going to be +1. Our R group here and the carboxylate in the backbone of the amino acid, they are both going to be negatively charged, which means the overall charge for aspartic acid at pH 7 is going to be -1. And lucky last is lysine. So lysine here, again, looking at our backbone, we have +1 for our quaternary ammonium group. We have our -1 for the carboxylic acid. And with this R group, because it is an ionizable R group, this quaternary ammonium group is going to be +1. So overall, the charge is going to be +1, -1, +1, which is going to give an overall charge of +1. So when we're looking at these three amino acids at pH 7, glycine is going to be neutral, aspartic acid is going to be -1, and lysine is going to be +1. Now, let's have a look at the next one. So what we're looking at here is essentially the same question, but what we've done is we have changed the pH. Now, generally in these types of questions, if I am asking you to calculate the charge for either an amino acid or a protein at a particular pH, I will always give you the pKa. Now, by that, I mean I will either A, provide to you the pKa, or B, give you a means in which you could calculate the pKa. So I might trip things up and give you, say, the Ka, for instance. Given that it is a pH of 14, this is a very, very, very alkaline solution. So it's quite clear that almost anything that can be deprotonated will be with respect to our amino acids and proteins. So let's have a look at the first one here. So I've drawn the backbone looking at glycine. Now, this is how we drew it at pH of 7. But we have a problem because this is not a pH of 7. We are now at a pH of 14. So what will change? Well, if we look at our carboxylic acid, can that become more deprotonated? No, that is as deprotonated as it's going to get. Our R group, well, our R group is non-ionizable, so nothing can change there. But what about our quaternary ammonium group? Yes, that can be deprotonated, and that is indeed what happens. So instead of NH3, that is going to turn to NH2. Looking now at our other two amino acids, looking at aspartic acid, well, again, we're looking at our three main groups that can be ionized. Our carboxylic acid, our quaternary ammonium group, and our R group. So looking at our quaternary ammonium group over here, that is going to be deprotonated. And of course, the carboxylic acid and the R group, which is also a carboxylic acid, they're all going to be deprotonated. Lysine, I'm sure you guys get the drill now, they're all deprotonated, especially because this is just so alkaline. So, and our quaternary ammonium group has lost that positive charge, and with lysine as well, it has also lost that positive charge in its R group down here. So, when we are calculating the charge, again, looking at glycine, our quaternary ammonium group over here, that is going to be neutral because it doesn't have that hydrogen there anymore. Then looking at our carboxylic acid, or in this case carboxylate, it is going to be -1. And our R group here for glycine is non-ionizable. So that means that the overall charge is 0 minus 1, which is going to be -1. Looking at aspartic acid, we've got our quaternary ammonium group that is going to be neutral in charge. Our carboxylic acid, that is going to be -1. And the R group is also going to be -1. So when we are calculating the charge, 0 minus 1 minus 1 is going to be -2. Now, what is important to keep in mind here is when we are looking at the quaternary ammonium group, the carboxylic acid, or the R group, they can only lose or gain one hydrogen, which means when we are looking at, say, our quaternary ammonium group over here, then it can either be as NH3 plus or NH2. It's only going to be moving plus or minus one hydrogen. So please keep that in mind. Looking at our lucky last one here with lysine, again, our quaternary ammonium group here is going to be neutral. It's going to be 0. The R group is also going to be 0. And our carboxylate over here is going to be -1. So the overall charge is 0 plus 0 plus -1. It's going to be -1 overall. So that is how, by looking at the structure and taking into account the pH, we can determine the structure and charge of our amino acids. Okay, so let's increase the difficulty slightly. Let's have a look at our amino acid, but instead of choosing such strong pHs, instead of saying just pH 1 or pH 7 or pH 14, let's sort of make these changes a little bit more gradual. So this question is saying to determine the net charge and the prominent form of aspartate at a pH of 1, 3, 6, and 11. So what I'm going to do here is just make a small note to myself just to sort of keep in mind the structure of aspartate while I'm working through this question. Now that I've drawn the aspartate just at the top there, again, just to keep it in mind, what I'm going to then need to do is use our pKa's. So when we are looking at this table has been derived from Leninger and is available on the Moodle course page for you guys. What we are going to do is observe what our amino acid is going to look like at each of these pHs. Now, let us start off with pH 1. Again, this is very, very, very strongly acidic. So looking at a pH of 1, it is safe to say that everything will be protonated. But how can we be sure? Well, let us look at this table. And to be specific, I am looking here. What this value tells me, again, it's the pKa. So remember, when the pH equals the pKa, 50% is protonated and 50% of the molecules are deprotonated. So what this tells me, looking at our pK1 here, so this would be of our terminal carboxylic acid group over here. What this tells me is that at a pH of 1.88, 50% of them are going to be protonated and 50% are going to be deprotonated. Now, if we want to be quite technical in terms of what is actually happening here, it would be a partial dissociation, but we're not going to go into that. What I want you to do, seeing as we're just starting with a lot of this stuff, I want you to make a couple of assumptions. The first one is, if the pH is less than the pKa, so in other words, the environment is more acidic than the pKa, then I want you to presume protonated. If the pH is greater than our pKa, I want you to assume deprotonated. And of course, if we have our pH equal to the pKa, then I want you to be assuming 50% protonated and 50% deprotonated. Now, as I've already alluded to, this is not strictly speaking truth. It is a little bit more complicated, a little bit more of a sliding scale and not so cut and dry. But as we're starting off, this is fine. So looking at a pH of 1, what we are going to be assuming is, again, looking at our pKa values over here, it's going to be completely protonated. This means observing our 3 main groups. What we are going to be seeing here at a pH of 1 is that everything is going to be protonated. So reminding ourselves the question, determine the net charge of the predominant form. So what would be the net charge of aspartate at a pH of 1? Well, again, looking at our quaternary ammonium group here, this would be +1. Looking at our carboxylic acids, this would be neutral, which means that the overall charge is going to be +1. Now let us have a look at how it's going, at how aspartate is going to look at a pH of 3. So at a pH of 3, we're starting to see some things change. So we need to compare our pH to our pKa values. So at a pH of 3, our pK1, or the pKa of our carboxylic acid here, the pH is greater than the pKa, which means this one here is now going to be deprotonated. Comparing our pH to pK2, which is our terminal quaternary ammonium group, it has a pKa of 9.60. So again, would this be deprotonated? No, it wouldn't. This is going to remain protonated. And looking at our R group, it is 3.65. Now, again, in the real world, this would be partially deprotonated, but we're going to keep it nice and simple here and essentially make the statement I said before of if the pH is less than the pKa, then it's going to be protonated. If it's greater than, it's going to be deprotonated. And what we can see here is that the pH is still less than that pKr, so this is going to be remain protonated. So our terminal quaternary ammonium group is going to be +1. Our terminal carboxylic acid is going to be -1, and our R group is going to be 0. So the overall charge of aspartate at a pH of 3 is going to be neutral. The next one is at a pH of 6. So looking at a pH of 6, again, we've got a couple of things changing. Our carboxylic acid here, 1.88 versus a 6. Note the pH is greater than the pKa, which tells me it's going to be deprotonated. The next one is our quaternary ammonium group. 9.60 versus a 6. The 9.60 is greater than the pH, so that means it's going to remain protonated. Our R group, however, is going to be deprotonated because 3.65 versus 6, that means that it is going to lose that hydrogen and become deprotonated. Looking at the charge, we have -1, -1, and +1, which means the overall charge here is going to be -1. And lucky last is looking at the charge at a pH of 11. So at a pH of 11, looking at our quaternary ammonium group, it has finally been overthrown. It can no longer hold on to that hydrogen, which means it's going to then change to NH2 and be neutral in charge. So we're going to be 0, -1, -1, which means the overall charge is going to be -2. And that, guys, is how we can look at and use these pKa values to determine what charge or how the amino acid is going to look at various pH ranges. So guys, thank you very much for joining me today. I'll see you in the next video.